There is a multi-billion-dollar micropropagation industry worldwide. Some of that is in Ireland. There have been up to 6 micropropagation companies in the Republic. And the reasons for this is because it's possible to produce plants very quickly through micropropagation. There are advantages and disadvantages of micropropagation I'm going to discuss. There are different methods of micropropagation, and the method can be dictated by the choice of explant. I've already covered the media that may be used, but it's very important to talk about different stages of micropropagation involved with stage 1, sterilisation, stage 2, multiplication, and then once you've actually got many plants, you need to go through stages 3 and 4, which is rooting, hardening off and transfer to the greenhouse. So the advantages and disadvantages of micropropagation. The great advantage is speed. So you get roughly a 10-fold increase every 2 months for many species of plants. It can be much greater than this, but as a rule of thumb, a 10-fold increase every 2 months. So it's possible to produce 10 to the 6 plants from a single starting plant in 1 year. I'm currently involved in micropropagation of potato, and I'm investigating a method for micropropagation in liquid media, and it's possible to get a 6-fold increase every week through micropropagation. So it's very easy to produce very large numbers of plants. The second advantage is that it's axenic. So provided the original explant is free of contamination, the resulting plants will all be uncontaminated. So this includes both viruses and bacteria. The third thing, it is clonal propagation. So plants are clonally propagated, so they're free of variation. So if you have a crop which is by its very nature clonally propagated, for example potato, it's very easy to clonally propagate potato in culture. And if you start off with a single uncontaminated plant, then you can have very large numbers of uncontaminated plants at the end of the process. One of the disadvantages of micropropagation is that it is expensive. Now you may think that 15 cents per propagated plant is relatively cheap, but if you compare that, for example, to Sitka spruce, where, I don't know, maybe a matter of cents per Sitka spruce seedling. If you produce an equivalent through somatic embryogenesis, then you're talking about maybe 30 cents per explant as compared to maybe a cent or 2 or 3 cents. From a historical perspective, micropropagation was first carried out in the 1960s with orchids. So there is a problem with orchids that conventional propagation only produces 1 plant per year. So basically the plant produces what's called a protocorm, and you can split this protocorm into 2. But through micropropagation of protocorms, it's possible to produce 1 million plants per year. So you produce a corm, which is basically a swollen stem, you chop it up, and each one of these will then develop into a number of other corms. You continue this, and then at the end of the process you'll need to mature them. Now there are different methods of micropropagation. More than 95% of all commercial micropropagation is through axillary branching. So production of protocorms, regeneration of protocorms, is a method of axillary branching. The great advantages of axillary branching is that it is genetically stable, so basically you get out what you put in, and it is simple and straightforward. So you just chop up the protocorms and allow them to get larger. If you have a shoot, you simply chop up the shoot into what are called nodal explants. So any explant that has a leaf on it will have an axillary bud in the axil of the leaf, and the axillary bud can be stimulated to grow out and give you a new shoot. For some species of plants, it's either more efficient to produce plants who have a shoot formation or it may not be possible to produce plants quickly through axillary branching. So a good example of efficiencies in adventitious shoot formation is St. Paulia. An example of where shoots are produced through adventitious shoot formation because it is not possible to do it quickly through axillary branching would be coffee. So it can be efficient, but it is possible, it's prone to genetic instability. So that's one major disadvantage of adventitious shoot formation. And finally, somatic embryogenesis. It's little used as a method for micropropagation, but it is potentially phenomenally efficient. So for example, I could meet the needs of Ireland and the UK for Sitka spruce through somatic embryogenesis in a couple of shake flasks, provided I can actually get them to grow up and mature in the shake flask. Axillary branching is very simple and straightforward. You take a shoot and you basically chop it up into nodal cuttings, which I've actually shown on the left there with the dotted lines around them. So you use shoot tips and axillary buds. People tend not to use shoot tips because they tend to become genetically unstable, so we tend to use these things here, axillary buds. So the bud will grow out to give you a new shoot, and basically you just chop that up. So it's very simple and straightforward. So once you've actually decided on an explant, you're going to need to sterilize it. You need to put it into a media that's got sugars in it, or some carbon source. It's got mineral salts in it, it's got vitamins, it's got hormones. All these things will support the growth of bacteria and fungi, and the bacteria and fungi will overgrow the medium or the explant unless they're removed, and the way that we remove them is through sterilization. Now you may get away with just sterilizing straight off, but it's usual to include some kind of pre-treatments to clean up the explant. So it may be you transfer the plants to a greenhouse to reduce endemic contaminants rather than trying to take something straight from the wild, from a forest. You force the outgrowth of axillary buds so that these are relatively clean. You use some kind of washing protocol to reduce or remove endemic surface contaminants. You may treat them with detergents before you actually sterilize them, or you may give them antibiotic treatments to try and knock down the contaminants before you sterilize them. But in any plant tissue culture protocol, you will very often see pre-treatment steps. You need to be able to identify these. Okay, so uses of detergents. Air bubbles on the surface of the explant, and I've shown this here, will protect whatever's underneath from the sterilant. So we've got loads of sterilant in here. Here is the leaf surface. Inside the plant is generally sterile. So all the contaminants tend to be on the outside. So if you've got air bubbles which can form around epidermal hairs and epicuticular waxes, then they will actually protect whatever's underneath, the bacteria and the fungi, from the sterilant, which is around here. So you need to get rid of them. So you need to mix in order to reduce air bubble formation, and you can use detergents as wetting agents to reduce the surface tension of the waxy cuticle on the leaf surface and increase wetting. For sterilants, there are three principal ways to kill off surface contaminants. You've got an oxidant action, you've got active halogen, and you've got heavy metal poisoning. And it is possible to actually use powerful chemicals such as conch sulfuric acid. I did try conch sulfuric acid as a sterilant, didn't work very well, but I have heard of it being used on seeds. Now, your sterilant will have one or even more than one of these actions: oxidant, active halogen or heavy metal. And there's always a trade-off between killing the surface contaminants and killing the explants. And as far as possible, cut surfaces should be protected. So for example, when I'm sterilising internodes, I will either make the internodes very large so that the ends get killed off, but I trim those off anyway. Or I would cap the ends with wax in order to protect them. So, sterilants used. Sodium hypochlorite. Hypochlorite ion has an active halogen. And you can see it's got this oxygen here. So, sodium hypochlorite and calcium hypochlorite are found in bleaches. So they have an oxidizing action. You know, if you spill bleach on yourself or your clothes, your skin or your clothes will be bleached white. So they have a dual action of oxidant and active halogen. There are better alternatives to sodium hypochlorite and calcium hypochlorite, things like sodium dichloroisocyanurate, which maintains levels of active halogen at physiological pH. Sodium hypochlorite and calcium hypochlorite tend to only produce active halogen at high pHs. So hydrogen peroxide has a very strong oxidant action. It's a very clean sterilant in that it breaks down to water and molecular oxygen. So you don't need to worry about chlorine and hypochlorite ion kicking around after you've done your sterilisation. And then there are these heavy metals. Mercuric chloride is used for explants that are difficult to treat. So for example, forest species tend to be difficult to sterilise. So you find for protocols using plants taken from the field, things like forest trees, will tend to use things like mercuric chloride. And they're a bugger to get rid of. They tend to persist in your cultures, so that they take a lot of washing to get rid of them. But once you've actually got your mercuric chloride, there are very few firms will actually dispose of this. So it's very difficult to actually get rid of it. So I tend not to use it. Silver nitrate is a heavy metal that will kill off bacteria. It's also useful. Remember when I was talking about the effects of ethylene? Silver ions will actually bind to the ethylene binding protein and inhibit ethylene activity. So if you've got a problem with your a big ethylene response in your explants, if you sterilise it with silver nitrate, you've got this dual action of heavy metals killing off the bacteria, but also the silver ions binding to your ethylene binding protein and preventing the heavy ethylene response. And you'll see that they're used at different concentrations for different times. And reason for this is that you have this trade-off between concentration and time. So you can use high concentrations for short times, short concentrations for high times, but there's always this trade-off between killing off the bacteria and killing off your explants. Antibiotics are rarely used since they tend to be bacteriostatic, and once you stop using them, the bacteria can then overgrow the culture. There are horror stories of people micropropagating 1 million plants, and when they went to transfer them to the field, they took out the antibiotics and the bacteria overgrew the explants. And there are no antifungal compounds that have been proven to be innocuous. So it's a numbers game. You start with a lot of explants, you sterilise a lot of explants, and you hope from this you get a few clean explants. Okay, the second stage is multiplication. So typically we're making nodal cuttings so you remove the inhibitory effect of the shoot apex on bud outgrowth. Gibberellic acids, this plant hormone, can be added to promote etiolation and cell division, especially in dwarf species or in species that tend to form rosettes. Cytokinins can be added to increase bud growth, and they will tend to antagonise oxidant effects. But this multiplication stage here is the most labour intensive part of the whole process. If you go to micropropagation companies, you'll see banks of laminar flow cabinets with individuals actually doing the multiplication work. They're very labour intensive. A lot of it nowadays tends to be outsourced to China, India, Vietnam, because labour's cheap. Stages 3 and 4 involve transfer to the greenhouse. So you're growing, you're multiplying these explants in a very rich media, high levels of sucrose, absolutely no necessity for the plants to photosynthesize. They're grown in a high humidity environment, absolutely no necessity for them to regulate the stomatal opening or prevent water loss by having waxy cuticles. So absolutely no necessity for them to have roots to take up water. So you need to root the plants by either dipping in an auxin solution or by using a medium that contains auxins. So auxins will actually promote root growth, and auxins are necessary for root growth and root initiation. The vast majority of roots in plants are actually produced adventitiously in the pericycle, which is the region immediately underneath the endodermis, which actually surrounds the core of the root. So we're exploiting a natural phenomenon. And then you need to progressively harden the plants, increasing the light intensity to get them to photosynthesize, reducing the sugars, inorganic salts and humidity, weaning them off these plant cell culture conditions. And you have to remove the medium completely prior to transplantation to prevent contamination. It's possible to micropropagate by adventitious shoot formation. I've already mentioned St. Paulia, and it can be phenomenally efficient. You can get 3,000 plantlets produced from one leaf. One plant might have 10 leaves on it, so you're talking about being able to get 30,000 plantlets produced from one source plant. In some other species, for example coffee, you need to produce this unorganized mass of cells called a callus prior to producing adventitious shoots. If it's possible to go directly from the explant, produce adventitious shoots directly from the explant, then you should go that way rather than actually producing callus. Callus tends to be genetically unstable, so you tend to get things like deletions, insertions, translocations, chromosome duplications, polyploidy when you go through a callus phase. And these are undesirable things in plant cell culture. Basically, when you're micropropagating, you want to get out what you put in. For control of organogenesis, the ratio of cytokinin to auxin is critical. If you've got roughly equimolar concentrations of auxins and cytokinins, then you tend to get a callus. If you've got high concentrations of cytokinin relative to auxins, then you tend to get adventitious shoots. If you've got high concentrations of auxins relative to cytokinins, then you tend to get roots. So it's possible to control rooting and adventitious shoot formation by the concentrations of auxins and cytokinins that you add to your tissue culture media. I need to talk about plant hygiene. So basically you get out what you put in. So if your plants are infected with viruses, then and you micropropagate them, then all of your micropropagated plants will be infected with viruses. The pathogens affect the yield, and for this reason there are very strict phytosanitation requirements for import of plants. So you need to get rid of viruses and bacteria, and once you've got rid of them, then micropropagate from your clean stocks. For elimination of viruses, there are a number of ways of doing it. One of them is through heat treatment. So I'm talking about growing plants at 37 degrees C rather than 20 degrees C, which they'd normally be growing at. And under those conditions, the plants actually grow faster than the viruses, and you can end up with regions of the plants that are free of viruses. Now, you can end up with quite large regions of the plants, so I'm talking about several centimetres, but for things like potatoes, you may end up with millimetres, maybe even only hundreds of micrometers that are free of viruses. So a way of actually getting rid of the viruses is what's through something called virus meristemming. So viruses are transported from cell to cell through plasmodesmata, and they're also transported throughout the phloem in the vascular tissue. But the apical meristem, especially where the plants have been heat treated, may be free of viruses. So there's a trade-off between infection and survival. So I've said that the meristems may be free of viruses. When I was thinking about doing a postgrad, one of the things that was offered to me was a postgrad looking at virus elimination in potato through meristemming. And what they were talking about was actually producing meristems of about 100 micrometers in diameter. If you go much above 100 micrometers, you have 100% virus infection. You go much below, let's say 80 micrometers, the meristems don't survive. So there's this trade-off between infection and survival. The other thing you can do is that you can assume that not all cells in the plants are infected. In some species of plants, they actually deliberately infect them with viruses to produce variegated plants. And in your variegated plants, you can see you've got green bits and you've got white bits. The white bits are where the viruses have actually killed off the cells. But if you can get adventitious shoots formed from the green bits, they can actually give you virus-free shoots. So, for elimination of viruses, you can start with virus-free plants. So, potato, for example, is naturally grown in cooler regions of Ireland. So, for example, up in Donegal, up in Scotland. And these will be naturally free of plants because the aphids which transmit the viruses don't thrive under cold conditions. Or you can heat treat plants to end up with meristems that are largely free of viruses. But at any stage you're going to have to test the viruses, and then for your virus-free stocks you can put them into your micropropagation cycle.